Hey guys, welcome back. So today we are going to attempt another question of the IBO 2013 year. Let's get started. Bacterial count of a liquid culture can be determined by different methods. Now, what are those methods that they are talking about? First, the cells can be counted under a microscope. So you take a small sample, then you put it under the microscope, and then of course you can count it. Okay. Second, you can use the Beer-Lambert's law. So what is Beer-Lambert's law? You take a cuvette, you put it into a spectrophotometer, and you pass the light. What happens is the bacteria or any sample, in fact, would absorb some of the light. Why? Because that's the property of the substances that we are made up of, right? So a limited amount of the light would be detected here, and this absorbance that happens is a function of how much of the substrate we have. Okay, so in case of bacterial cultures, we usually use A600 600 nanometers because it does not damage the cells in our let's say DNA or whatever. We don't want any damage just to happen. That's why you use A600. Now, the absorbance of 1 corresponds to 8 times 10 to the power 8 bacterial cells per ml. Okay, so they have given you some information. Third, several dilutions of the culture can be plated on agar, and the colonies can be counted to calculate the number of colonies forming per units per milliliter. So what you do is you dilute it as they have shown in this figure. You take 1 ml and then add 9 ml of just the media. Now this is called serial dilution, right? Because you are doing it again and again. So you again take 1 ml, add 9 ml, 1 ml, 9 ml. So every time you're doing it, you're multiplying it or diluting it by a factor of 10, right? So 10 times 2 times 3 times 4 times 5 times 6. So at the end, you have 10 to the power 6 dilutions there. And each time you plate them. What do you do? So you mix the solution well. You take a loop and a plate, and you just, there are different ways of plating, but you do this, right? And you wait it out for the bacteria to form colonies. You count the colonies, and the number of colonies that there are, that means that's the number of bacteria there must be there. Okay, because you have diluted them a lot of times. You're hoping that a single bacteria will give rise to a single colony. Okay, so pretty simple. Let's read the questions now. Okay, a culture with A600 is equal to 0.1 and a doubling time of 30 minutes will reach 4 times 10 to the power 8 cells in less than 2 hours. Okay, now the doubling time is 30 minutes. So 30 minutes, the concentration of the bacteria is just going to double. If the concentration goes to double, that means the absorbance will also change. So 0.1 is what we have at 30 minutes. Okay, and if we have an absorbance of 1, that will be equal to 8 times 10 to the power 8. Now the bacteria would double again at 60 minutes, so we'll have 0.2 absorbance. Once again, the bacteria will double at 30, right? So 1 hour, 30 minutes. And in this, we can have the absorbance of 0.4. And once again, the bacteria will double at 2 hours. And at that time, we'll have the absorbance of 0.8. Okay. Now, you see 8 times 10 to the power 8 bacterial cells, that is when you have 1. Okay. So at what point of time will you have half of it? That is what the question is asking, 4 times 10 to the power 8, right? So at 0.5, right? Now you clearly see that 0.8 is achieved at when it hits 2 hours and 0.4 when it hits 1 hour 30 minutes. So 0.5 will be somewhere in between. We can see it from the data. So a culture will reach the limit in less than 2 hours. Yes, that's correct. So you'll mark it true. Okay. Counting colonies on plates gives a smaller estimate of number of bacterial cells than counting cells under a microscope. Well, if you're counting colonies on the plate, what will happen? We are counting the colony forming units. You have to understand that colonies will only be formed by living bacterial cells, right? Dead cells, they will not form colonies. But when you're counting the cells under a microscope, living or dead, you'll count them all. So counting colonies will of course be a smaller estimate. Okay, so this again goes true. Third question, using the plate giving the most accurate results from the figure, the culture X is estimated to contain 1.6 times 10 to the power 5 CFU per ml. Now first you have to figure out which of the following will give you most accurate results. So these are the plates. As a rule of thumb, we tried to go for dilutions that are 10 to the power 3, okay, or they should not be between 300 to 30. If you're going for a very less number of CFUs, you are likely to introduce error by chance. And if you're going for more, you stand a chance at failing to distinguish the two separate colonies. So the best number is 160 as per R. So it's 10 to the power 3 dilution and between 300 to 30. So there is 160 colonies if the dilution factor is 10 to the power 3. So you simply use unitary method. If the dilution was 1, then you'll have to simply multiply, right? So you'll have 1.6 into 10 to the power 5. That is exactly what the question is quoting for you. Okay, so this also turns out to be correct. Now coming to the last question. The question says, when repeating the plating of dilution F many times, some plates will show colonies. So what is F? F is 10 to the power 6 dilutions. Now let me remind you, from the data above, you have 10 to the power 3 dilutions as 160 bacteria per ml. Now in 10 to the power 6 dilution, what do you have? You'll further reduce the count. So 16 by 10 to the power 6. Now this is not straight maths I'm doing. It's just I'm doing it methodically rather than applying the formulas, right? So okay, sorry, not 6, 3, because the difference is of 3. Okay, so that comes to be 0.16 per ml. So in 1 ml there is 0.16. Now it's just like turning a dice. So what is the probability that you'll have the face of 1? If you throw a dice, it's 1 upon 6. That means if you throw the dice 6 times, at least one of the times you'll get 1. So the probability of getting 1 bacteria is 16 times 6 is equal to 96. So if you do plate more than 6 times, you'll have achieved a probability, or you must get at least 1 bacteria. Okay, so when repeating the plating of dilution, at least some plates will show colonies. So yes, if you do it more than 6 times, you will get a colony. Okay, so this was the question. I would say it was fairly easy, but do keep on trying and solving more and more questions. Also guys, there's this book, NCERT map, I talked about in the last video as well. Do check it out on Amazon. If you find it funny, then share it with your friends. Also let me know in the comments if you need some more help. Thank you.